Hello, everyone. Thank you for attending today's webinar, Our Gaps in Your Clinical Pharmacology Program, Jeopardizing Your Drug's Approval, presented by Certara. My name is Joan Wiseman, and I'll be moderating this seminar. I'd like to start off. We have a remarkable panel of speakers, and I'd like to start off by introducing them. Julie Bullock is the Senior Vice President and Global Head of Clinical Pharmacology and Translational Medicine. Justin Hay is the Senior Director of Clinical Pharmacology and Translational Medicine. Kritika Shetty is the Director of Clinical Pharmacology. Blair Osborne is the Senior Director of Clinical Pharmacology and Translational Medicine. Paola Coppola is the Director of Clinical Pharmacology and Translational Medicine. And Eva Jill Berglund is the Senior Director of Clinical Pharmacology and Regulatory Strategy, all at Certara. You can read their full bios on the left side of your screen by selecting the Speakers tab. Just a couple notes before we dive in. There are handouts available for download. You can access those by clicking the Handouts tab on the left side of your screen. If you want closed captions during the presentation, you can access those from the bottom right corner of the video player. This webinar is being recorded, so you will be able to watch it on demand within 24 hours after we conclude here today. We would love to hear from you. If you have a question at any point during the presentation, use the Q&A tab on the left side of your screen and submit your question. At the end of the presentation, the speakers will answer as many questions as time permits. All right, we're ready to begin. Julie, please go ahead.
Thank you, Joan, for the introductions. And thank you to everyone for attending today's webinar. My name is Julie Bullock, and I'll be moderating and introducing my colleagues. I've been consulting for about 9 years, and prior to my role as a consultant, I was at the FDA for 10 years as a clinical pharmacology reviewer and team leader. I'm delighted to be moderating today's session with my colleagues, and we look forward to sharing some common clinical pharmacology issues that we have seen in our roles as consultants and in our former roles as regulators. At the end of today's session, we hope that you will be able to understand the key clinical pharmacology hurdles that have been seen in submissions that can impact approvability, and we will also provide some strategies to mitigate these program gaps so you can avoid these problems in your development program. Before I hand this over to my colleagues, I wanted to share an overview of our clinical pharmacology regulatory strategy team and services here at Certara. Our experience is unmatched, with 7 former FDA, EMA, MHRA clinical pharmacology reviewers and 35 global clinical pharmacology scientists with scientific and regulatory experience supporting a variety of client programs. Our clinical pharmacology regulatory strategy team has provided unique insights into clinical pharmacology program strategies. We've helped with submission and document messaging and have assisted with resolving many stalled development issues, among other services. I am very proud of the dynamic and talented clinical pharmacology team we have here at Certara, and I'm a firm believer in the value that access to former regulators can bring to your development programs. I'll now turn it over to my colleague Justin Hay, who will provide an overview of the importance of addressing clinical pharmacology issues based on his published work while he was at the MHRA.
Thank you, Julie. So what I will start off with is first saying that the regulatory agencies have a wealth of knowledge and occasionally publish on the reasons that lead to delays during the NMA or MMA procedures. So the first study I wanted to present is about the one from Tufuri, who looked at withdrawn or refused applications over a 7-year period at the EMA. And for major objections in efficacy, approximately 20% were based on PK issues or bioequivalence. However, the paper that Julie mentioned was a paper that I wrote while I was at the MHRA and the European Medicines Agency, where we looked at major objections across a 5-year period. For new active substances, the vast majority were due to issues describing the dose exposure response relationship. However, other issues were due to characterizing the PK in the target population, analytical methods, ADME, and interactions, all topics that we're going to explore in more detail during this webinar. Similarly, for orphan products at the European Medicines Agency, the determinants of success included several clinical factors, but also identified the need for sound dose finding data as well as as it was an important consideration. When looking over a decade's worth of NME applications assessed by the FDA, they categorized first cycle review failures. Clinical pharmacology issues were included in several of these, including inappropriate populations, inappropriate doses, or confounding conmeds. Today, we're focusing on clinical pharmacology issues that may lead to delays and questions during the review that may impact approvability. Some of these are unavoidable and which may need to be managed, while others, there are issues which are avoidable and which can be planned around. So before we move on, please note that we're going to provide several examples. However, we're going to only identify medicines when needed. So the first topic is PK in the target population, which was one of the topics identified as the main issues in clinical pharmacology major objections paper that I'd wrote about. So it's vital to characterize and understand the PK in the target population. It may seem quite an obvious one, but this is sometimes missed. There are cases where PK in the target population has not been sufficiently investigated, and in the first case on the left, PK was limited and not characterized properly. In this case, the application was refused. In another case of a transdermal patch, skin adhesion, which could have had an impact on permeation and PK, was only investigated in healthy volunteers, but not properly investigated in the target population, which was elderly. In other cases, and similar to the first case, there was scant PK available in the target population, such that only single dose data were available and limited PK data was collected in the pivotal study. Ultimately, this and other issues led to the application being withdrawn. Lastly, on the right is an example where PK data was collected in the patient population in one region, but that was not representative of the population in the region where it was applied for, and the application failed. Ultimately, limited small populations are generally not justification for not collecting PK in the target population. Also, if using alternative populations, sometimes the final population differs from the initial studies where a lot of PK is collected, but it still needs to be representative of the target population. In many of the cases above, population PK was used to try and patch proverbial holes in the application. However, this was identified by the assessors, and it's always important to use PK analysis appropriately. Thanks. So now we'll move over to Krithika, who will discuss issues related to dose justification. Thank you.
Thank you, Justin. Good morning, everyone. My name is Krithika Shetty. I'm a director of pharmacology at Certara, and I used to be a clinical pharmacology reviewer at the US FDA. In this next section, I'd like to speak to you about the importance of adequate dose justification in your development program. The ultimate goal of any successful dose optimization program is the identification of a dosage of a medicine that achieves adequate, if not optimal, risk-benefit balance. In order to have the smoothest review of your dose justification at the time of market application submission, it is important to consistently develop and clearly describe the dose story of your drug. In other words, it is important to clearly explain how the registrational dose of the drug was selected and how data from the registrational study or studies continues to support the appropriateness of the proposed recommended dose. Any robust dose justification would ideally clearly identify how the dose is supported by the totality of evidence available from the development program. In other words, it would explain how it's supported by integrated information available from the 5 key areas that are highlighted on this slide. That includes translational studies, clinical PK data, clinical PD, clinical safety and efficacy, as well as dose and exposure response relationships of safety and efficacy with drug dose or exposure. A few key notes in order to help sponsors successfully do this are identified in the block on the right. First and foremost, it is essential to have robust clinical dose ranging studies. Modeling and simulation can help fill some gaps, but it cannot completely salvage missing or significantly lacking clinical dose ranging assessments. On the topic of modeling and simulation, for most programs, we find that it is extremely informative to start these activities early in development, to iteratively update these analyses at key dose decision points or at key regulatory milestones. And importantly, it is crucial to heed the messages of these early modeling and simulation analyses. Structured presentation of dose arguments in regulatory submission documents is key. One of the key misconceptions is that clinical pharmacology reviewers at various health agencies are only interested in reviewing PK/PD or ADA data. This is not true. Clinical pharmacology cares about clinical safety and efficacy data as it relates to dose and does review this material. And therefore, it is important to cover these aspects in ClinPharm-related review sections. Another recommendation we'd like to make on this topic is to think about maintaining a dose justification white paper for each program, which can be updated as development proceeds in order to maintain clarity and consistency of dose justification arguments throughout the history of the program. Finally, it's also important to seek timely feedback or to engage with regulators in a timely fashion such that adequate information is available to inform the discussion, but also such that any feedback received remains actionable as it relates to your clinical plans. I would like to reemphasize that inadequately justified dose can negatively impact the overall risk-benefit assessment of the drug, not just from a clinical perspective, but from a ClinPharm perspective, but also from a clinical or approvability perspective. And I'd like to provide a case example of this on the next slide. This is a case example for what we call drug A, which was a monoclonal antibody intended or in development for the treatment of ulcerative colitis. The first market application submission for drug A was met with a complete response by the FDA due to the modest overall treatment effect, which was further confounded by the potential for a suboptimal dose selection by the sponsor. A key element of this decision making was the exposure response analysis, which indicated that over the range of exposures at the proposed recommended dose, clinical benefit increased with increasing exposures, with no apparent plateau in the exposure response curve, which suggested that the dose selected was potentially not maximizing clinical benefit. The second resubmission of the market application for this drug for the same indication was discussed at the Gastrointestinal Drug Advisory Committee, or the GI-DAC, where the FDA continued to recommend a complete response or non-approval for this drug. However, given the need for additional treatment options and the favorable GI-DAC vote, the drug was ultimately approved. However, the approval was associated with a dose optimization BMR to evaluate further higher doses of the drug in this indication. This case example clearly demonstrates how inadequate dose optimization early in a development program can significantly add on to both development time as well as resource later in a drug's life cycle. With that, I'd like to hand over to my colleague Blair, who will take us through the next few sections of the presentation. Blair?
Thank you, Krithika. Good morning. My name is Blair Osborne. Until June of this year, I was a senior reviewer in the Office of Clinical Pharmacology, Division of Cancer Pharmacology at the U.S. Food and Drug Administration. Currently, I'm a senior director in clinical pharmacology at Certara. Formulation. I was going to start with discussion of formulation changes. I tend to place drug development challenges into two categories. First, there are those that are fully under your control. As a company, you plan if, when, and how these changes will be addressed. Second, there are those issues that arise that are outside of your control. You don't get to choose whether these are addressed, but you do control how they are dealt with. I consider formulation changes generally part of the first category. They're under the company's control. The dose form and formulation of the drug change over the course of development. This is expected. The stage of drug development, when new formulations or dose forms are introduced, can impact both the timeline and the number of studies needed that need to be conducted. Planning for a change in your drug presentation can reduce the impact on your development timeline. The lot of drug used in your pivotal trial must be representative of your commercial drug that you propose to market. In the approved filing, the primary evidence of safety and efficacy come from that pivotal trial. If the lot of drug proposed for marketing is appreciably different from your study drug, then the safety and efficacy of your commercial drug has not been demonstrated. Approval will be delayed or denied until this has been done. The amount of data to support your formulation change increases the later in development the project is. Changing a dose form during phase 1 or early in phase 2 may only require demonstrating comparability. The bar is much higher after the pivotal trial, as you now need to demonstrate that the new form of the drug is expected to produce both the safety and the efficacy in alignment with the clinical trial form. These are two actual examples. On the left is a diabetes drug, where the drug used in the clinical trial was analytically different from the to-be marketed formulation. The difference resulted in a complete response letter being issued. The sponsor was able to address the issues raised and to resubmit the NDA. However, the timeline was prolonged. An approval was eventually issued, but it took 24 months from the initial filing and a substantial amount of effort on the sponsor's part. On the right is a proposed biosimilar. In this case, there were 3 major flaws, any one of which could have derailed the program. First, the proposed biosimilar did not meet biosimilarity criteria when compared to the reference product. Second, the lot of drug used in the clinical trial did not align with the proposed commercial batches. And finally, in an unusual circumstance, the data on the reference product itself did not agree with the historical bioanalytical data on the same reference product, calling the analytical methods into question. This BLA was withdrawn. Modifying dose forms over the course of development is completely acceptable. Making the effort to strategically plan this can minimize the impact on your development timelines. My apologies, it didn't move forward. Okay, I'd like to switch gears and talk about QT prolongation and its impact on drug development. I consider QT prolongation an example of the second category of drug development challenges. While you cannot control whether the drug has the potential to cause QT prolongation, you can control how to present the risk management of this. QT prolongation is an intrinsic effect of some drugs. It is not possible to eliminate this through planning. QT prolongation represents a safety risk that can cause development programs to be discontinued. Approvals may be delayed, and additional studies may be required. Regardless of this, many approved drugs do have QT prolongation concerns. So while it's not possible to avoid the problem, it is possible to address it. A key to success is having a successful and timely approval by identifying the risk and planning how to manage and monitor it in patients. This is a recent example of the approval path for a drug with QT prolongation as one of its toxicities. The original NDA was filed in 2018. After an initial review within FDA, it was presented at an Oncologic Drug Advisory Committee, or ODAC, meeting. ODAC is composed of experts from outside the FDA who propose who provide additional perspective on questions posed on proposed drugs. In this instance, the ODAC members were asked to vote on a single question. Do the results of the pivotal trial demonstrate that treatment with this drug provides a benefit that outweighs the safety risk for the patients? For this drug, ODAC voted no, indicating they believed that the potential benefit did not outweigh the potential risk. The benefit was modest, and there was an increased risk of cardiotoxicity, including a risk of cardiac death. The NDA was subsequently withdrawn. A new NDA was submitted in 2022. This submission contained a new trial with a different patient population, a different dosing schedule, and new proposed plans for the management of the QT prolongation. This altered the risk-benefit evaluation, and the NDA was approved this year. From here, I'd like to move to bioanalytics. The relative robustness of bioanalytical work is wholly under your control, but it takes considerable effort to make sure that things are done well. Bioanalysis is often an overlooked part of drug development. Having solid bioanalytical work may be taken for granted, but it really should not be. The reported concentrations, they serve as your basis for exposure response analysis, population PK. The data is used not just to select doses and schedules, but also to identify populations at risk, like the elderly, and to make appropriate dose modifications. If you do not have complete faith in your bioanalytical data, how can you support these critical decisions? The clinical pharmacology reviewers at FDA do review bioanalytical data. As part of the review, they may consider several areas, like whether the method has been appropriately validated for the conditions of use in the study, and whether the implementation of that method for study sample analysis was done in accordance with the validation conditions. This slide provides examples of bioanalytical flaws. The flaws range here from not following SOPs to not adhering to regulations to outright fraudulent behavior. The takeaway message here is that if the bioanalytical data have to be discarded, an entire trial may have to be repeated. The impact on approval timeline is very difficult to avoid in this case. Not every bioanalytical flaw is a disaster. Many flaws in bioanalysis can be corrected with some additional work. However, even a correctable flaw can result in a delay or a denial of approval if it's not dealt with. These can be avoided wherever possible. If method validations and sample analysis reports for pivotal trials are not submitted as part of the marketing application, the results can be a refusal to file. In an FDA analysis conducted in 2018 indicated that there were 66 refusals to file based on clinical pharmacology issues over that 9-year period. So while it's not common, this does occasionally occur. Even once an application has been accepted for filing, problematic data may not be considered during the application review. These data are simply set aside until corrections or additional data actually make them relevant. Some commonly encountered issues are not getting samples analyzed within the long-term stability window, mismatches between the validated matrix and the study matrix, poorly selected validation ranges for assays that don't reflect the actual study data. These issues can be corrected, but they likely require additional data or additional analyses, resulting in delays for approvals. As an example, an NDA was submitted in 2015, but the submitted bioanalytical work was considered to be unreliable. A complete response letter was issued, and the sponsor was required to develop and validate a new bioanalytical method to address the deficiency. The NDA was amended and resubmitted in 2017, and the approval was issued 6 months later. But the flaw resulted in a considerable delay of approval for this original 2015 submission. Thank you. I'd like to turn the presentation over to my colleague, Paula Coppola, who will now present with a discussion of concerns in mass balance studies.
Hello everyone, thanks Blair. So I'm Paola Coppola and I'm a director of clinical pharmacology in Certara and I'm a former reviewer at the MHRA and EMA. We'll now talk about the human mass balance study, which is one of the most informative in the clinical pharmacology package for new drug application, and it's crucial to understand the clinical PK properties of new chemical entities and to drive the clinical development journey. So, what information will you get from this study? The human mass balance study is usually conducted with radiolabel compound, and along with the non-clinical mass balance study, provides essential information on the exposure and elimination of the parent and metabolite. For regulatory purposes, there are two major objectives. The first one is to identify and quantify the circulating drug-related material and to investigate whether there are metabolites contributed to the drug safety and that may require standalone non-clinical characterization. The second objective is to get an understanding of the fraction absorbed and to characterize the drug clearance pathways. So ideally, more than 90% of the dose should be recovered as total radioactivity in the excreta, and more than 80% of the recovered radioactivity should be identified. And common issues in the regulatory review are about the portion of the total radioactivity characterizing plasma, or they can be about the plasma samples not collected long enough to quantify the metabolites profiling, or about the excreta not collected long enough to have a complete understanding of the elimination pathways. Which administration route should be used? Ideally, an IV dosing should be used or even for oral products, and this will be very important because only the IV provides a reliable estimation of the absolute bioavailability and of the contribution of different elimination pathways, like, for example, the biliary secretion. So here in this slide, we have three examples of common cases related to the mass balance challenges that may lead to major review issues or delay of marketing authorization. In the first case, the clinical pharmacology package does not include a human mass balance study. So the lack of this study should always be justified and supported by providing robust and valid justification. So let's see which justification might be acceptable. For example, for molecules such as monoclonal antibodies or endogenous substances, it may be not acceptable to not conducting a human mass balance study if the ADME properties are well known from previous studies. Or another example can be for the antibody drug conjugates products, as the human mass balance study might not be feasible for these compounds, but still it will be needed to have an understanding of the human elimination pathways. And this can be done, for example, through the evaluation of the excreted metabolites in the urine and feces from early clinical trials or from in vitro and animal studies. Another justification could be that the data show that more than 90% of the dose is recovered in the urine as unchanged parent and there is minimum metabolism, or if the data provides reassurance that there is negligible or no systemic exposure. Then, in some cases, it might not be possible to conduct a human mass balance study, for example, for safety reasons. So, in this case, if adequately justified, a bridge with the non-clinical, in vitro, or other clinical data without radiolabel compound may be accepted. However, if you don't have a full understanding of the elimination pathways, this will likely bring to strict label restrictions for co-medications, and so this is likely to be requested by the regulators. The second example here is about the timing issues. So the earliest you plan the human mass balance study in the drug development plan, the better. And this will be an advantage for you because a complete characterization of the elimination pathway may be time consuming, and quite often additional studies are needed before the clinical pharmacology package will be complete and ready for submission. So ideally, the human mass balance study should be conducted at least before the phase 3, because if the late stage studies rely only on non-clinical or in vitro information, you may not have a full understanding of the elimination of your drug. And this could either cause delays in the regulatory review or make the regulators asking for post-authorization measures, which are known as PAM in Europe, and this will likely cost you additional studies and possible label restrictions. The last example is about the unique metabolite. This is a specific situation where a metabolite is identified during the clinical development but is not present in animal tox species, or its exposure in animals is much lower than in humans. And so in this case, the regulators expect further non-clinical evaluation to investigate the potential toxicity of that metabolite. Um, Based on the FDA guidance, there are two possible approaches that can be used to address this issue. The first one is to identify an animal tox species that form that metabolite at least at the same exposure as in human, and then investigate the drug toxicity in that species. However, this might not always be possible. So if it's not possible to identify an animal species that forms that metabolite, the second approach will be to synthesize the drug metabolite and then directly administer it to the animal for safety evaluation. And although this can bring challenges and possible complications, this is very important for safety evaluation. So what if this is not conducted? This will likely affect the review of your data and lead to requests of additional studies or post-authorization commitments and label restriction. So now that I've talked about the possible changes regarding the human mass balance studies in regulatory submission, I would like to hand over to my colleague Eva Berlon. Thank you.
Thank you, Paula. So my name is Eva Schild Berglund. I've been with Certara for 4 years, and before that, I was a ClinPharm reviewer at the Swedish MPA for over 20 years and working a lot under the EMA umbrella. I will happily continue on this theme and talk about the value of knowing your drug elimination and how essential it is for your ClinPharm package in impacting the risk benefit. So the mass balance study is essential, actually to such an extent that as Swedish reviewers, this was the only study that we would look into by default, even in cases that we were not the rapporteurs in EU. The knowledge is central. The information about the primary elimination pathways coming from the mass balance study, together with the in vitro data on the drug's ability to be a substrate to specific enzymes or to transporters, and then follow up with a DDI study or a pharmacogenetic study confirming and quantifying the role in vivo, sets the stage for one of the key ClinPharm tasks, the identification of situations at risk for relevant over or under exposure. The impact of many intrinsic and extrinsic factors can be predicted. Here, PBPK can be very useful. So high level, PBPK is a modeling approach where we simulate up a drug behavior based on a physiological model of the human body. The simplest situation is to predict DDIs with other inhibitors and inducers, followed by pediatric drug exposure, informing your pediatric drug development. But as science is growing, we are starting to predict the impact of renal impairment, of hepatic impairment, disease states, obesity, pregnancy, and so on. This information can replace studies, it can support labeling, or it can be used to design your ClinPharm development and individual studies. So this is not just an exercise aiming at DDIs. The mechanistic information is also key, enabling predictions of many real-life situations. Not having this information impacts the risk-benefit assessment. The impact depends on the therapeutic window and the ability to catch situations of overexposure by monitoring. It also depends on the expected target population and therapeutic situation, for example, if you have polypharmacy. A situation where you're not able to predict the victim DDI is difficult to resolve by general DDI statements in the labeling. Usually, insufficient information in organ impairment is easier to handle in labeling, but in real life, this depends on the clinical need. Unknown or difficult to manage DDI risk can also impact the product's viability post-approval. This is important to remember. The cases to the right here are all drugs that were withdrawn from the market due to severe DDI consequences. We have also had other situations over the years where we have not been able to predict DDIs, for example, before the transporters were sufficiently known. Here, this caused severe safety issues post-marketing. To illustrate the impact of the risk-benefit assessment, I've included two examples below, one on DDIs and one on renal impairment. The first case, drug A, illustrates how lack of knowledge of major enzyme and transport involvement, not enabling victim DDI predictions, can impact the risk-benefit assessment. This is an extract of the public EU assessment report, the EPAR. This drug was not approved, not only because of this, but it did have an impact on the risk-benefit. The next example, D, is a narrow therapeutic index drugs with 90% renal excretion. Here, the exposure starts to increase already at mild renal impairment, the same decrease in renal function that we often see in the elderly. The safety in the elderly population was indeed questioned. This was part of the ground for refusal in the first marketing application, which was rejected. In EU, you can ask for a re-examination after rejection, and this was done for drug D. Here, the product got approved, but renal impairment was not allowed. In the elderly, investigation of renal function was mandatory before starting treatment, and the renal function was monitored throughout treatment in all patients. A PAM, so the correspondence to the US PMR, was issued to investigate a suitable dose in mild renal impairment. In conclusion to these slides, elimination knowledge is key. It sets the stage of the ClinPharm development plan and enables valuable simulations of situations at risk, informing both your clinical protocols and your labeling. Lack of this information also impacts the risk-benefit of your application. I'm coming back to the comments from Blair. Not having this information is something you in most cases can avoid. So don't put yourself in a situation not having it if you can. Instead, understand your elimination in time and benefit from your knowledge when you're planning your development ahead. With this, I will give the floor to Julie for closing comments.
Would look like for a clinical pharmacology program. I think that we all understand that we should be ideally getting some PK in our target population so that we can understand and characterize the pharmacokinetics and the exposure response. We would obviously like to have a good and robust dose justification that has totality of evidence, dose ranging, as well as early modeling and simulation to put into that dose justification. Formulation, the key here is that timing does matter, and when you switch your formulation and what you need to provide from a bioequivalence or a bioavailability perspective will heavily depend on whether or not you change formulation during IND or after submission. For QT issues, in this instance, I think preparation matters. So we can't not collect the data. We do need to address it for almost the majority of assets that we will be developing. But I have found that early planning and getting QT information and early clinical studies is almost always far better and will serve you better than waiting to address QT later in development. For bioanalytics, oversight matters. So make sure that we have a good validation parameters and we have trial analysis within those validation parameters, or develop the necessary data to expand the validation. And for human ADME, many people don't like to do these studies because they're expensive and time consuming, but I do think that they are probably one of the most important studies in the clinical pharmacology armamentarium so that we can really understand our assets and the metabolites. And I've definitely seen and been involved in a fair share of consulting for clients that have delayed ADME studies until phase 3 or post phase 3, and a lot of surprises have come up. So in summary, our overall recommendations, I find that a bare necessities approach to your clinical pharmacology package will often provide you inadequate information for approval as well as labeling. I have worked in this space for long enough that I can say that if your approach is to skirt the line of a refuse to file, the overall NDA or BLA application may not be viewed as highly as a program that definitely gives the regulators things that they need to inform labeling for patients. The other thing is that clinical pharmacology is a fairly broad discipline, and it requires us to collaborate with pharmacometricians, biopharmaceutics, DMPK colleagues, as well as our clinical and statistical teams. So it's very important that we don't work in isolation. We involve all of these teams as well as regulatory. Planning for early development is really important, and set that up early. This should be a living document. It should be very broad. It should have things that you plan to do by end of phase 2, by phase 3, and post-approval. I don't think that there is a good excuse for just ignoring clinical pharmacology development plans. Yesterday, I was on a phone call where the client was like, "We're just moving so fast, we haven't done anything for clinical pharmacology." And I'm like, well, that's great, but I don't think the regulators are going to agree with you. Timely regulatory and actions. This example was actually an early asset for phase 3 where they have had no interactions with the regulatory authorities for clinical pharmacology from IND to phase 3, which is not something that you should attain for. So with that, we will start with the panel. So the rest of my colleagues can come back on screen. And thank you to those who have already put questions in the question and answer box. So I'll go ahead and actually start with one that has come up quite a bit. I've noticed a few times is absolute bioavailability, and whether or not approval will be delayed if there's an absolute bioavailability absence or whether or not that is pending. So I will answer this from the US FDA perspective, but I'll look to Justin, Paola, and Eva to answer it from the EMA perspective. But absolute bioavailability is not something that is necessarily a refuse to file issue for applications. It definitely does help understand your bioavailability between oral and IV. But if there are no plans to develop an IV product or an oral product for your asset, that is not something that is absolutely required for your filing. Relative bioavailability obviously is something that we may need to do for formulation changes. So Blair, Paola, or Eva, any questions there?
No, I mean I can talk from the EU, and I would say that I would completely echo what you said. I think that the agencies are very aligned. It's never mandatory, or I can't think about any situation where it would be mandatory to do an IV or bioavailability study, but it can be helpful in certain scenarios. But it's never a must to have this study.
Yeah. Let's see here. Let's go to QT. So Blair, this one's going to be for you. If you don't have any non-clinical hints that the product might have a QT risk, do you have to perform QT studies nonetheless? I'm assuming they mean in the humans in a clinical study.
Yeah, that's a good question. I think that it depends on what you mean by non-clinical and what you mean by QT study. You have to have a certain amount of non-clinical data. For instance, you're going to need to have a hERG assay in vitro, etc. You're going to need some of that information. And once you've gathered up enough non-clinical information, even if it is all negative, you're still going to need to collect some information in the humans. That does not commit you to a full QT study at this time, but you're going to need to collect, particularly in your first in-human study, samples at both the initial dose at Cmax and then at steady state Cmax, so that you can have triplicate ECGs and understand QT there. Once that is done, if you think that you have enough information to build something, to build an argument, you can submit it to FDA and say, we'd like to waive our QT study because of all of the following things. Occasionally, that is successful if you are one of the drugs that there's a bunch of information on. However, if you are a new drug, you're probably going to have to go back and acquire additional information before you can move forward with your development.
Very good. Thank you, Blair. So Krithika, you're up next. We have a couple questions that are coming in about dose justification. So one is asking about the dose justification white paper and where we can find this paper.
So there isn't sort of a template for it that we can hand to you. It's just a recommendation for something individual companies develop internally. A nice resource to help with setting this up is I want to call it a worksheet that the Office of Clinical Pharmacology at the FDA has released recently in the context of Project Optimus in oncology drug development. It's available on the FDA website. It contains the overall major subheads as they relate to dose optimization that are applicable more broadly regardless of therapeutic area outside of oncology as well. So I'd point you to that document, and certainly this is something that any trained clinical pharmacologist should be able to help you set up within your organization.
Yeah, very good. And then the next question, Krithika, for you for dose justification is with regards to rare diseases. So in this instance, the person who's asking the question is saying, what if in a rare disease we have no dose range finding study and one dose tested in the target population? Could modeling and simulation be applied, including other populations, to explore exposure response with common endpoints?
Right. So there's a few different elements to think about regarding this question. We, of course, appreciate the challenge with full-fledged, if you will, dose optimization exercises in rare indications. But with specific reference to what I think are the parameters of this question, in terms of exposure response for a safety perspective, it's certainly useful and very informative to leverage information available in other indications, right? So ER safety from other indications can be quite informative. But when it comes to efficacy, exposure response relationships for efficacy, borrowing them from one indication to the other in order to justify dose, it kind of depends on target biology and whether or not we can build a mechanistic or a paper-based argument to support the notion more broadly that exposure response relationships with efficacy in the different indications are not significantly different. If the answer to that question is yes, we can build that sort of argumentation, then it may be feasible to use well-designed dose optimization studies and other indications and exposure response analyses thereof to justify your dose in the rare indication. However, if you're unable to build that sort of rationale, then it becomes more essential to look at exposure response and dose response in the rare indication in question. Now, when it comes to building that exposure response in the rare disease, it is sort of challenging to use data only at one dose level, which is also likely a small sample size to build that exposure response. And especially something about this question made me think this might be an oncology indication in the context of Project Optimus, that idea of extrapolating exposure response at one dose to surmise what might be happening at other doses may not be acceptable in an Optimus universe, at the FDA at least. So I'm happy to discuss any more specific parameters of that question, but this is sort of a broad overall answer. I hope that was helpful.
Very good. Thank you, Kritika. So Paola, I'm going to move to you. So we have a question that came in that says, if a drug is being developed as both IV and oral, should you run mass balance studies for each?
Okay, so I think this is a very good question. So I think it depends on the PK properties available from other studies. So it depends on the specific ADME data that you got, for example, from the non-clinical species, because having only the oral or the IV could bring you to missing some important information on your drug elimination, because if if there is, for example, first pass metabolism, then only the IV will help to provide an estimation of the contribution of the elimination pathways to the drug clearance. But in other cases, if, for example, you expect to have a large fraction of your drug as unchanged in the feces, you might have a important role of the biliary or the gut wall secretion to the elimination of your drug. And so in that case, the oral will be important. So it's difficult to just say, yes or not. I think it depends on the specific ADME properties of your compound. But ideally, having both administration route will give you a full understanding of the real elimination of the drug.
Yeah,
I hope this
absolutely.
answered the question.
Yeah, no, that's great advice. So we have another question, which I've seen this a couple of times actually. A case where the mass balance study in healthy volunteers shows that the circulating radioactivity is mostly accounted for from the parent. But the study in renally impairment healthy volunteer studies show that the metabolite exposure is higher than the healthy volunteers. So, which supporting data might be requested by regulators in this space where there's maybe discrepancy in metabolite exposure between healthy volunteers versus patients with renal impairment, or healthy volunteers with renal impairment?
Okay, yes, thanks, Julie. So I think this will be a specific situation that can be a bit tricky, because usually the human mass balance study should be conducted in healthy volunteers, as you said, but it can be that in some cases a metabolite becomes more important in special populations. So in this case, the regulators would expect further evaluation of that specific metabolites and the contribution on any toxicology issues. So I think that additional non-clinical toxicology studies should be provided. And when it comes specifically to the renal impairment, the contribution of this metabolite to the pharmacological activity should be evaluated in that specific population. And here the modeling and simulation, like the exposure response relationship, could be very useful to provide that reassurance.
Very
I
good.
hope this answers the question.
Yeah, no, absolutely. So I have a question actually about PK delays during pregnancy. So this actually could go to you, Paula, as well as you, Justin, given your pediatric experience and the experience there. But are there any cases of delays in the approval of a drug for use during pregnancy that potentially was attributed to they say PK issues, but I'll go broader, clinical pharmacology issues. Yeah. Any experience there for either of you?
So I can try to start, but from my experience, I
Maybe,
haven't
Paula, you could just
came across
explain some of the clinical pharmacology issues that we need to consider when we're dealing with pregnancy, maybe just high
yeah.
level. Yeah. Okay.
Yeah, okay, no, that's good. Yes, yeah. So when we're dealing with use of drug in pregnancy, we need to consider a lot of changes, physiological changes in the pregnant women that can have an impact on the PK properties of a drug. So it's really important to have a very good understanding of the absorption, distribution, metabolism, and excretion of the drug, because, for example, pregnancy can have an impact on the activity of some enzymes involved in the elimination pathways of the drug. So, for example, if the drug is a substrate of of some CYPs, specific CYPs, and we know that pregnancy can increase the activity of those enzymes, then you can expect to have lower exposure of that drug in pregnancy, and this could bring to possible effectiveness issues, or in other cases, pregnancy can have an impact by increasing the activity of those drugs, and then decreasing the activity. So in that case, an increasing of in the systemic exposure can bring to safety issues. And other considerations should be done in terms of the protein binding. So if the drug is highly bound to the plasma protein, particular attention must be done to the free fraction, because it's expected to change in pregnancy. So, yeah, there are a lot of factors to be considered.
Yeah, absolutely. Well, thank you, Paula. Thank you for that. So I'll move a little bit over to the DDI, the land of drug interactions, with you, Ava. One of the questions we've received is, if elimination knowledge is key, how would regulators look at a situation where we have not been able to identify the involved major enzymes or transporters despite in vitro work, human ADME, and follow-up DDI studies?
Yeah, so sometimes there are difficulties in identifying the metabolizing enzyme, and maybe you start by your candidate enzymes that you have in vitro, and also based on your mass balance study, going on to make DDI studies, and then you actually don't see the expected impact of your inhibitors. So being in that situation, you have to go back and look again on your mass balance data to see what kind of metabolic reactions do we actually have, which enzymes might be involved. And when you have done thorough work in vitro, looking at metabolite formation, trying to map up your metabolism schedule that you see in the mass balance, if you still don't identify your key enzymes that are transporters, um, there comes a point where you just cannot provide any more useful information, because in the end, we want to know this information to be able to predict DDIs or sometimes predict the impact of special populations, but mainly DDIs. And there, if you're coming in a situation where you can exclude that all the enzymes that react to predict DDIs based on that they are not involved, I mean, then you have done what you can, and you just have to live with that. But of course, it's the regulators will not ask you for more, because you can't use the information in the labeling. But of course, you might have a situation where things might happen post marketing. But yeah, there comes a point where you just have to give up.
Very good. Thank you, Ava. And one last question I'll send to Justin. So, what can be done to mitigate some of these issues that we've discussed today, from a clinical pharmacology perspective, from your perspective?
Yes, thanks, Julie. So actually, one of the findings which we looked at in the paper was to look at whether scientific advice had been given for the European side of things. And of course, in generics, this generally wasn't the case, but for, of course, the new molecular entities and the complex biologicals, scientific advice was given quite commonly. So that's one route is to be in contact with the regulatory agencies, whether it's through the European Medicines Agencies, through the national agencies on an individual level, or of course through the FDA, through their meetings. But of course, the other major thing is to have a good clinical pharmacologist on your team. It
Yeah.
makes they can identify, and as you said, it's multifactorial, so they will bring in the right person as well, generally, as
Yeah.
needed.
Absolutely. Great advice. I know that a lot of biotechs usually cobble together a clinical pharmacologist with a non-clinical toxicologist and a statistician, but that usually doesn't make a clinical pharmacologist. So definitely important to make sure you've got that discipline on your team. So it looks like we're out of time. So I'll hand it back to the organizers at Fierce to spend some closing remarks.
Thank you all for attending this Fierce Pharma webinar and submitting so many fantastic questions. Special thanks to our panel of speakers for participating and for Certara for presenting today's webinar. A recorded version of this webinar will be available for you to access within 24 hours. You can use the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.